Great. Well, thank you, everyone. We are very excited today to have Dr. Tara Melvini from Expression Systems, an Advancion company. And this is a further in Thomson's lecture series on educating our customers and our scientists and scientists that use our products, use Expression Systems products in different methods that are to be used in cell culture. Today, we are lucky enough to have Dr. Tara Melvini, who's the president of Expression Systems, discuss the discovery, the power of insect baculovirus expression, efficient production and scalable solutions for protein, AAV, and vaccine production. And after that, I will follow up with some of Thomson's solutions and just working with Tara for, I think, Tara, the last 20 years.
So,
It's like a day or two, but just enjoying, an expert in her field and someone who's really advanced insect expression. And as all of us in expression would say, sometimes we don't know enough about insect and baculovirus. And that's why we had Tara join us today for just a great explanation and showing some of the possibilities that we can do with Advancion's or Expression Systems system. So Tara, if I can turn it over to you.
great. Thank you so much for that wonderful introduction, Sam. Next slide. Thank you. So, to click, pop it. So we're here to talk about the Baculovirus Expression Vector System. What is it and where can it best be utilized? The Baculovirus Expression Vector System was discovered in the 1950s. It was discovered in an academic lab that was studying crop pests. And what they had noticed was that these insects were being infected by a virus that was producing a very large amount of a particular protein that the virus uses to spread from insect to insect. And the scientists at the time looked at that and said, wow, if we could just tell the virus to produce the protein we want to express instead of this protein, we could generate a whole lot of our recombinant protein that we want to make. And so that's sort of what kicked off the Baculovirus Expression Vector System. It was adopted pretty quickly by the academic and research areas because it was so effective. And then it made the leap into commercial applications by being a very good way of producing animal vaccines. And there's a number of those on the market. Of course, once you demonstrate efficacy of a production system, the human vaccine market got involved, and that was started with the launch of Cervarix, a human vaccine for human papillomavirus. Another application that was adopted very, very early on was expressing an array of proteins or modifications of those proteins for drug discovery efforts. So now we've got sort of two tracks. One is utilizing it as a tool to express proteins to further research in other applications, and then also as a tool to actually produce products of commercial interest. And one that's been in the news a lot lately is expressing AAV for gene therapeutics. So that was very exciting expansion of the field. Next slide. So one of the biggest adopters of the insect cell and baculovirus system was producing GPCRs to understand their structure. And GPCRs, as you may or may not know, are a very large class of proteins that are very, very important in a number of signaling areas. So they're a great target for drug discovery and looking at interactions of how other molecules work within the cell. And that was really where expression systems got involved in this application space, is our customers that were producing these GPCRs for their crystallization efforts. And we're very excited to share that GPCRs, the bulk of GPCRs that have been resolved were actually expressed using insect cells and baculovirus. And we were a part of those and are the most highly cited media manufacturer in these academic publications because of our involvement with these products. And here I'm just listing 3 very recent high impact journal articles that continue this work today. Next slide. So now that we know why we're here, why do I get to talk to you about it? So Expression Systems was started in 1997, and the focus really was the high throughput protein expression for drug discovery. That's who we targeted our initial product for. And we have a very collaborative relationship with our customers. We want to help our customers utilize the platform to their best ability, and we learn so much from our customers. And that really helped us become the leading experts in the baculovirus expression platform in the industry. It also pushed our efforts from a research media manufacturing company to where now we're a best-in-class cGMP cell culture media manufacturer. And we couldn't do it without strong relationships with our customers. So we're highly appreciative of that. We are registered as a medical device manufacturer, and we are ISO 13485 certified because of our relationships with our customers. And they'll call up and say, hey, how do I do this? I don't know. Let's do an experiment. We do an experiment together. And that evolved into our contract services arm of our business, where we will do work for our customers. They might be wanting to try out the system and they just don't want to put in the infrastructure efforts without knowing it's going to work. They might just be at capacity. There's a number of reasons why people will come to us to help them do their work. And as we became more and more comfortable and familiar with this and the opportunities arose, we've expanded to a cGMP CDMO for early stage preclinical and clinical products. We've been part of the Advancion Corporation since November of 2022. Next slide. So why the baculovirus and insect cell expression platform, and what is it? One of the main advantages is it's very easy to use. And as I'm showing you right here, there are 3 main components of the platform. There's the insect cells themselves, there's the cell culture media that you grow them in, and then there's the DNA that you use to produce the recombinant baculovirus that's going to express your protein of interest. It's also a very safe system. It's a biosafety level 1 system. So you don't have to take a lot of precautions for people that are utilizing the system. It's highly productive. We'll show some data as we move along to convince you of that. It's economical, particularly in comparison to the HEC 293 system. And there's a low barrier to entry. If you're already growing mammalian cells, you can just change your incubator temperature to 27 degrees C versus 37 degrees C. We have customers that just use a shaker on their benchtop. These cells will grow at ambient temperatures, although it's not the most productive, but if it's good enough for the customer, that's great. Very high precedence of use. I am continually amazed by the applications that our customers have extended this to. Our customers are really the experts. There's so many things that you can do with this system. We couldn't possibly reiterate all of them. And that's why we really try to develop these collaborative relationships. So it's a 2-way learning experience. And you can use this system at the bench all the way up to commercial applications. A notable example of a commercial application of this system is Novavax's COVID vaccine that you may or may not have heard about, and flu block vaccine by Sanofi. The next slide. So here we're going to talk about if you've decided to take that leap into the insect cell expression space and see what you can get out of it. How do you choose your upstream manufacturing platform? How do you choose your cells, which media you want to use, et cetera? And so for your platform considerations, it's really important to look at the end use of your protein product. Are you going to just be producing a protein for a research application, crystallography, as a candidate drug target for your candidate drugs? That's one aspect. Or are you do you have aspirations of developing a clinical product. And that's going to tell you what system you want to be working in. Many of your top priorities are going to be how much time does it take to develop this? What are the quality attributes of the components I'm working with? Which one's the easiest to use? What's your production environment? Again, are you on the bench? Are you in a clean room? Do you need to scale this production space? Expression Systems offers liquid media packaged both in the 1 liter bottles all the way up to a 200-liter bag in a drum. For quality compliance down the road with your product, what quality attributes do you want from your cells and your media and your DNA? And then, of course, the overall cost of the system. And so as we move through this talk, we'll be talking about different cells and different medias. Next slide. We're going to focus on 2 upstream manufacturing platforms today. Everything that I speak of as we go through this has been done in both of these media formulations, and it really lends itself to what is your application that would ask you to decide what media you want to pick. So our ESF 921 platform is our most popular product that we sell. It's our flagship product. The fact that this media formulation came out in 1997 and is still our best seller really speaks to what a robust media this is. It was developed for structural biology. It's ideal for that. It was developed with the intent of high throughput protein expression applications. It's a protein-free, serum-free media formulation. And we also provide research-grade Spodoptera, the SF9 or SF21, or Trichoplusia, the TNE cell lines. Now moving over to our ESF Advanced CD platform, this is our latest entry into the media space. This is our next generation and easiest to use product from Expression Systems. It was developed to be really seamless in the adaptation from another media formulation into advanced CD. We know that's a real barrier to entry to our customers when they want to move their cells, but they don't want to have to go through a time-consuming adaptation process. It was developed specifically for the AAV manufacturing market. It's ideal for that and for virus-like particle manufacturing. But we're also finding that it's so robust a media product that people are adopting it for ease of scale or better protein expression. And because it's chemically defined and because it was developed based on all the knowledge that we've gained over the last 25 years, it can improve your reproducibility and accuracy of your scale-up process. Like I mentioned, it's chemically defined. It's also animal origin free. And you can use it with our research grade SF9 or TNE cells. And we also have a rhabdovirus-free cell line, the SF9 RV-free cells. Next slide. So I'm just going to mention these briefly in case one of your considerations is further clinical manufacturing. The Spodoptera cell lines that are commonly available have an endogenous rhabdovirus in them. This is a Spodoptera rhabdovirus. It's not super concerning, but it is a rhabdovirus. And one of the most famous rhabdoviruses out there is rabies. So the regulatory agencies are, we don't want any rhabdovirus in your clinical product, please. And now there's a number of commercially available rhabdovirus-free cell lines out there. And so now they're asking, if you are particularly in AAV manufacturing, if you're going to be producing this, we want you to use a rhabdovirus-free cell line, or you're going to have to do all of these virus clearance assays. And so the way that we manufactured our RV-free cell line is we did single cell printing. And so you can see circled in the green circle, a single cell that was deposited into a single well of a 96 well plate. And then we grew out these clones. And so that's how we derived this cell line. Just wanted to mention it, that we have a master cell bank that's been produced under cGMP and completely tested for use in clinical applications, should that be a consideration. Next slide. So we've talked about the components. How do you put them together and actually make them work? So usually when you're doing your protein expression, there's two big buckets of work. There's the upstream process and then there's the downstream process. We're not going to go into a lot of details about the downstream process, but we can, should you need help with that, we can help you with that when you get to that point. We're going to focus on upstream bioprocessing today. So the first step is to generate that recombinant baculovirus that's going to make your protein of interest. And you do that by transfecting your DNA into an insect cell, and out pops your recombinant baculovirus. You do this using Spodoptera cells. Trichoplusia cells are not indicated for this application, but you can use baculovirus that was produced in Spodoptera cells to infect Trichoplusia cells. It's supported by any Spodoptera cell line. We have a BestVac product that you can use to generate your recombinant baculovirus. We have other reagents like virus stabilization additive to maintain that virus stock. And we do all of our work in Thomson flasks or deep well blocks. And I'll show you some data in a bit. After you've generated your recombinant baculovirus, then you're going to want to go into the expression. I know that it's also referred to as a fermentation, so you'll hear me use those terms interchangeably throughout the rest of the talk. You just take your cells, put them in a flask of some sort with your media, and then you infect it with your recombinant baculovirus, and you express. It's as easy as that. It is a lytic system, so there's a defined time from the time of infection to the time of harvest. It's kind of a short scale. I think I need to bring that up for those of you that are used to like CHO fed batch cultures and stuff like that. It's not like that at all. It's just a very limited lytic expression. And you can broaden your application space with your cell lines from the Spodoptera cells also to the Trichoplusia cells, commonly known as T or high 5. And then there's other like novel insect cells out there also that are less commonly used. All this work can be done using either the ES921 or the ESF advanced CD media formulations. Next slide. So how do we generate a recombinant baculovirus? So you want to create your transfection complex to go into the insect cells so that they can produce it. In our case, we do co-transfection method. We use our BestVac products, which is a linearized baculovirus, and we, through homologous recombination, combine that with a transfer vector that has your gene of interest expressed in it. So you take your BestVac DNA and your plasmid DNA, and you put it in with some transfection reagent. That can be PEI, it can be a lipofection reagent, and you mix that up in a transfection medium. And we highly recommend this because it increases the uptake of the DNA into the cell, and can help in situations where you might not have the most clean transfer plasmid product. So you mix that up and incubate it for 20 minutes at room temperature. Our standard process in the past was we would then take that transfection complex and add it to approximately 1 million cells in the well of a 6-well plate, and that was cultured adherently. And we would incubate that for 4 to 5 days. And the output was 3 milliliters of what we call a P0 product of your co-transfection with a titer of 10 to the 5th to 10 to the 7th infectious units per mil. And this was produced with ESF 921. Well, we had customers who didn't like that yield. They thought that the volume was too low. They thought that the titer was too low. It didn't really compare with what they were getting from a back-to-back system where they're just doing the transfection and they don't have to rely on homologous recombination. And so we tried to add some efficiencies into the process. So we moved to the suspension method, and we used Thomson shake flasks for this process. We exchanged that 1 well of a plate to 20 milliliters of cell culture at 1 million cells per mil. We put the exact same amount of transfection complex into that shake flask, incubated for 4 to 5 days. And in this case, we yielded 20 milliliters of product with a much higher titer of 10 to the 8th or 10 to the 9th mils per mil. And this was sort of a game changer. It cut an entire amplification cycle out of the process and really sped the move to actually moving forward into your expression. And we can do this with all of our media formulations. the data that I'll show you is in either ESF 921 or advanced CD. You can't culture the insect cells adherently in the advanced CD media formulation, so we didn't do any of the adherent studies with that. Next slide. So what we are hearing from our customers is it's always about the efficiency. We want more faster now, right? We're all familiar with that edict from above. And as we moved from adherent to suspension, that immediately added efficiency. But we wanted to see if we could push it beyond the 20 mils in a shake flask. Can we go up to a more standard 50 milliliters per shake flask? And then also for high throughput, we started incorporating the deep well blocks from Thomson to do a bunch at once. And in this, so what I'm showing you is 125 ml shake flask, optimum growth flask, as well as the deep well blocks. And in the graph, we're looking at the infectious virus titers that we are getting out of these co-transfections. And in the first batch, we looked at the titer at day 4 and day 5 post transfection. As expected, day 5 had higher titers as the virus keeps producing. So we'll focus on those orange bars. In the 20 mils, we were at almost 10 to the 8th infectious units per mil. In the 50 mils, we were at 1E9, which was amazing. Again, we're using the exact same amount of DNA, so there's a cost savings there also. And then in the deep well blocks, we put 25 mils of culture in the deep well block and got close to 1E9 infectious units. Again, what I liked about the deep well block, and this is one of the experiments that I like because I got to do it myself, is I loved working in the deep well blocks. It was so convenient to just have 6 cultures open and you could add your reagents, and it was super fast, super easy, very efficient. We did see a little lower yield in the 20 ml shake flasks versus the 50. And as we've worked with more and more of the Thomson products, we realized it was likely due to the humidity conditions in our laboratory. We're in Davis, California. It's very hot. We have low humidity. And so we were having a lot of evaporation unbeknownst to us in the 20 ml culture. It wasn't dropping the volume so much as to be concerning, but it was impacting the yield. We've started playing with the low evaporation caps that are provided by Thomson, and we're seeing increased growth, and we expect to see increased yield with those flask caps. Next slide. Okay, so we've quickly gone through how to make your recombinant baculovirus. How do you use it? So you basically, again, back to ease of use with this product, you seed your production vessel with your cells and your media. It can be Spodoptera cells or it can be Trichoplusia cells. It can be a variety of volumes. We'll be talking about cultures that range from 50 mils of culture up to 5 liters of culture here. ESF 921 and advanced CDs support robust cell growth across a variety of flask sizes and volumes. And we have recent customer data that shows that ESF advanced CD can increase expression up to 6-fold. So that would also be a consideration when you're choosing your platform. But back to the ease of use, you put your cells in your flask with your media, you let them grow overnight so that they're in a nice logarithmic state of growth and receptive to infection by the baculovirus. Then you add your baculovirus, and you should do some titrations to determine your best MOI and really understand your platform. But basically just throw in some virus, let it incubate 2 to 4 days. That time is going to depend on how much virus you added up front. And then you harvest. Pretty easy. Next slide. So how does it work? I'm sure is your next question. And here we're going to start showing you some examples of protein expression. And here we're looking at membrane protein expression as measured by fluorescence intensity on the surface of the cell. This was performed by a third-party collaborator. And they were just looking at how does ESF advanced CD compare to a competitor's media that they'd been using in their laboratory. The bars in orange were produced using advanced CD, and the bars in blue were using the competitor's media. In both cases of protein A and protein B, you can see that the level of fluorescence intensity is higher, and so you had more protein expression using the advanced CD product. What was interesting to me was these two proteins were expressed using different promoters, and it's independent of promoter usage, the advanced CD worked better. Next slide. This customer also was producing virus-like particles, and again did the same sort of experiment, a head-to-head with the competitor's media and our media. If you look at the left-hand graph and the two bars in the middle, in green is the competitor's media, and in orange is the advanced CD. Head-to-head, same infection density, same MOI, and an almost fourfold increase in expression of the virus-like particles. Now, we found that this is a really robust media formulation that supports really high growth. You can get cell densities of Spodoptera cells up to 30 million cells per mil in the advanced CD media formulation. And so we had suggested trying a higher infection density and a lower MOI. And so they did that experiment. They infected at 5 million cells per mil with an MOI of 0.1. And you can see in that far right bar that they got extremely high levels of protein expression using that strategy. So they're just, it seems, well, it is simple. This is a simple system. You just increase your cell density and you increase your yields per liter. Easy as that. They took it one step further in the analysis, and they looked at expression at the per cell level. And you can see in the blue is the expression per cell in our competitors' media versus advanced CD. So it was a double whammy here. Not only did we increase it by the cell density, but the cells themselves are producing more protein. Next slide. So as I mentioned, this media was developed specifically for AAV expression. And here we were doing the experiments that were suggesting that if you can get better yields as you drive up the infection density. So following our standard processes, we seeded at 2, 3, and 4 million cells per mil. We let those cells go overnight for our standard procedures. And now we're infecting at either 4, 7, or 10 million cells per mil. We're producing AAV, so we have 2 recombinant baculoviruses that we have to add to the culture, and we add them both at an MOI of 3. So we're putting in a lot of virus here. And then we harvested it at 72 hours post-infection. It is a lytic process, as I've mentioned, and you can see in the lines on the graph that the viability has dropped over the course of the infection, as you would expect. And it's consistent between the 3 different culture conditions because we've controlled for the MOI. That's great. Everybody's excited when your cell cultures look the same, but we only really care about how much protein did you produce. So if you move over to the graph on the right-hand side, we're looking at AAV titers. And in this case, the titer was determined by GPCR. So we're looking at the number of genome produced per ml. In the seed density of 2 million versus 3 million, we saw a doubling in the amount of protein expressed. If you move to a seed density of 4 million, now you're looking at an infection density of 10. You're kind of pushing it there, and we didn't see any improvement in yield going that high. So we've really started using this seed at 3 million, infect at 7 million strategy for all of our AAV productions in this system. Next slide. So that was work that we did. People tend to be more comfortable with if the manufacturer isn't the only one doing the work, showing that this is working really well. And so we have some data from a customer who was screening different media formulations for their AAV production. So this work was done in shake flasks, and they were just looking at the AAV from the protein expression, not after purification. But what we were really happy to see is across 8 different media formulations, you can see in that second bar that the ESF Advanced CD was by far the best expressor of AAV in a head-to-head comparison. Next slide. So all the work that I had showed you previously was done in a 50 ml shake flask, pretty standard benchtop expression conditions. But we want to scale. We want to produce more protein than that. And so we work closely with Thomson, as Sam mentioned. We've been working together for like 20 years, and we've been using a variety of their different flasks to do scale up. So here we're looking at production at the 200 milliliter, 500 milliliter, 2.5 liter, and 5 liter expression conditions across an array of optimum growth flasks. I'm still, my favorite flask is still the 5 liter flask there. It's the third from the left. I really like the big boys, but I don't know, there's just something about that 5 liter flask that I've gotten very comfortable using it. It's I do like the wide mouth. It's easy to manipulate. You don't have to worry about bumping your pipette into the edge. So the flasks were just following our standard procedures, seed the culture and let it go overnight, infect, harvest 72 hours post-infection. If you look at the bottom graph, you can see that our seed densities are consistent and our infection densities are consistent-ish. Our protocols say culture overnight. There can be a lot of interpretation between morning and evening. And so that's what's accounting for the variability in the growth in the infection bars there. But in all cases, the cells grew very well in all different volumes and all different flasks. And then when we go to harvest, as you would expect, the viable cell density goes down because the cells are starting to die as a result of the lytic infection. But what's most important is the yield. And so if you move to the graph above it, in the two blue bars, we're looking at AAV titers, in this case measured by capsid ELISA in the 200 ml flask and the 5 liter, or the 200 ml culture and the 5 liter culture, and their equivalent. And so it's a very scalable process, both from the insect cell expression platform and the bioreactor container, the flasks that we're using for this expression. Next slide. So I hope in this whirlwind presentation that I have convinced you that the insect cell and baculovirus system is very easy to use and very effective in producing the recombinant protein of your choice. There's a multitude of proteins that can be expressed. It works for secreted proteins, intracellular proteins, and membrane proteins. It's fantastic. And we're here to support you in any of those efforts with our product line. We, as I mentioned, really focus on the cell culture media and then have the accompanying cell lines that go along with it. We also have molecular tools. I've mentioned BestBac. I didn't go into the details. There really wasn't time for that. But we do have products that you can use to generate your recombinant baculovirus, transfer plasmids, transfection reagents. And then we have other reagents that we didn't discuss at all, like our production boost additive that you can add after the infection's been established and will help you produce more protein. If you're interested in more information, you can go to our website, expressionsystems.com, or email info@expressionsystems.com. Also, this was a high-level presentation from Expression Systems, and we really liked to tailor our conversations based on the customer's specific needs. And so we can actually come out, I can come out, our other scientists can come out and do a technical talk or have a conversation and help you with your specific workflow. That's the end of my time here. So I will turn you over to Sam now for some information from Thomson. Thank you.
Great. Thank you so much, Tara, for this. Vic, if you can advance the slide. And we will have question and answer at the end. So just to give a background on Thomson, we are a business all based in Carlsbad and Oceanside area, and we are a diverse business. We have been doing for the last 20 plus years is making single-use products in collaboration with customers like Tara and like yourselves to improve expression and also clarification and purification. If you want to go to the next slide, Vic. So again, we're on our side, we are agnostic. We do like what Tara is doing in insect cells. We do work in HEC, CHO, and then clarification, and then purification, and then also for at the end for FPLC or HPLC or UPLC, we do have our filter vials for checking that. But whether you're growing plasmids or whether you're growing insect cells or other types of cells, we have the tools that you need. So if you can scroll forward. Vic,
I did, Sam. We're on the next
sorry,
slide.
yeah, it just doesn't show up. So we have over 48 patents. We have over 200 products we did. And last year we came out with the 7-liter flask. We've been working on the 6-well, and we'll show more 6-well products that we're developing for 2025 and 2026, just so that we have a mid-scale process. And again, we're looking to work with other labs who are looking for innovation and more tasks, more proteins. We don't always have the amount of people we need to do it, but how do we look to it and speed processes up and automate as much as possible? So if you can scroll to the next slide, Vic. So we look at it. I am a structural biochemist, and we've always looked at how workflows change. And just this, we've seen, my grandfather was a chemist, my father was a chemist. No one changed the flask until we looked at doing this from 1860 till about 2004. We always see Honest Abe, the honest truth. I wish I had Thomson flask back in 1861. We would never be using a vector or a cell line from 1860. So we wonder why people use the same tools that people in the 1860s use. That's where we adapted our processes for modern day. The optimum growth actually came working with JCIMPT, Ray Stevens out at Scripps, where we redesigned the flask because they wanted higher densities, larger volumes for what they were doing for membrane proteins. And that's where our initial work came from. If you can advance to the next slide. So again, what we did was make scalability from 96, 24, 6-well, and from 125, 250, 1.6, 5, 2.8 liter, 5-liter, all the way to 7-liter, where we can have volume up to 40%, 40 to 50%. The 5-liter and the 7-liter has worked well. And again, you're increasing protein amount and reducing our plastic waste. We're all going for a green initiative. And how do we make it simpler? And so if you can scroll forward, Vic. So again, we're looking at 4 times higher OTR, twice the volume per flask. If you can scroll forward, Vic. So this is some of Tara's data that she sent to us for PEGs in 2025. Again, just showing scalability between doing a 200 mil culture all the way to 5 liters for AAV. And she spoke more specifically. And what we like about working with expression systems is everyone may have their own projects, but expression systems has the expertise in insect cell work to move projects forward. And we really feel when it comes to protein scientists, you need the expertise. It's not just about the media. It's about looking at the whole system. And that's why we like working with expression systems. If you can scroll forward, Vic. So in this year, this is something new that I'm not sure everyone has seen. We did a 7-liter flask, whether it has a spout or just a 7-liter. Some of our customers are doing between 5 and 6 liters for insect cells. If you can scroll forward. So on the left, we're showing CHO transient, and we've done CHO stables, where we've done up to 5 liters. Start with 80%, 40%, and then 10% fill, 10% fill. And we'd like to thank Pfizer for being our beta tester also on insect cells, one of many, but they were able to do 6 liters in SF21, again, at 135 RPM at 25 millimeter orbit. So if you can scroll up to the next slide.
Mhm.
So something very interesting that came out recently that's on our website, we worked with Lundbeck. Lundbeck was able to show that they were able to do comparable data with 7 different proteins with a 7-liter flask with CHO and get the same comparative data to a 200-liter bioreactor that has pH and DO probes, and we got the same normalized titer. We got same VCD, and we also had a little bit less lactate than they did. And we maintain the same cell viability. So again, we are now seeing people use our 7 liters with between 10 and 20 flasks at a time, but to do 50 to 100 liters. This way, there's no need for a wave bag, no need for a small scale bioreactor. If you are going to PD and other processes, that might make sense if you're doing final process for a large scale. But we are seeing a lot of PD and early manufacturing and research go to our 7 liters as a simple tool. I mean, the bioreactor is about 20-fold more expensive for single use. It's also time to set up, where you can use our 7-liter and just drop it in the shaker. Next slide, please. So again, Lundbeck did a comparison with a Cytiva 50-liter, a Corning 5-liter, which they could only do 2.5 liters in, and a Thomson 7-liter with 5-liter. They got the same results between a 50-liter Cytiva bag that has DO and pH control to a Thomson 7-liter shake flask, which allows for more flexibility. And this allowed them to scale up with Thomson 7-liter flasks. If you want to scroll to the next slide, please. So again, our flasks are all designed to use in the same shaker. So you can go, this is an enforced shaker from 125 all the way up to a 7-liter shake flask. If you can go to the next slide, please, Vic.
Mhm.
So what does VCD and viability look like? VCD was equivalent in our 500 mil, 5-liter, and 7-liter. So everything was scalable. The nice thing is it's scalable results from our Thomson 125 to 7-liter. And this also saves time because you can skip sizes. You can go from a 125 to a 500 mil to a 1.6, 2.8, or 5-liter or 7-liter. So you can go forward, Vic. So just interest in time, they were able to do a PK/PD study from one Thomson 7-liter doing 4.3 grams after purification. So again, this speeds up studies. Every first to market, and this gives the PK PD very fast and show. If you can advance. Again, we looked at protein quality. We were able to get 50% more volume, but when you have a hard GPCR protein, you need as much protein as possible. We were able to get 8% increase in target proteins. So again, 50% more volume, so plus 8%. So when you're getting 10 mgs out and you can get another 15, 16 mgs, just allows for another assay, another cryo-EM. If you can scroll forward. So again, we do have spouts, so you can connect it for GMP, and we do have a transfer cap and a regular 7-liter. So if you can go forward, Vic. So what we're doing now in 96, 24, and 6 well, if you can go forward, Vic, just what processes we're developing. So we're going to go into our 6 well that Tara talked about between 20 and 50 mils at 225 RPM and 25 millimeter. If you can scroll forward. So again, we are and have been working with teams on a integrated lid. We're improving the process for that currently, and we're looking to grow 1 to 7 days, but we have proven we can do up to 50 mils. We're also building this with automation so that it is automation friendly. So if you can go to the next slide. So again, with SF21, we showed here, thanks to Pfizer, we showed we were able to do scalable between 25 and 50 mils. For SF21, we've done this for tinies and high fives. If you can scroll forward. So what does it work in? We wanted to thank Inforce for this. Their peg trays have worked perfectly. We've been able to do their 4-position and 6-position peg trays, and we can do this, or they have a full peg tray. They can do 24, and then if you do have a Cooner, there is a Cooner rack that can do 24 at a time, and that allows to do 144 constructs. We are working with automation to take the Inforce tray and drop it right onto robot decks. If you can scroll forward, Vic. So again, we're showing here that you can do 144 constructs, do 20 to 50 mils. This, for a lot of customers, eliminates tubes or TPP spin tubes, and then is a simple way for mid-scale. If you can scroll forward. We are also developing a universal rapid clear. This has no binding, so it provides rapid clarification with minimal sample loss. So if you can scroll forward, Vic. And we would like to thank the team who helped develop it at Genentech, and also with Ed Kraft at Leash Technologies. We show here with Villia, this is also with mammalian cells, where we showed that we had no loss from before, from afterwards, where again, great clarification, optimize of the purification, and it's ready to go. So if you can scroll forward. So we showed the same thing with leash technologies with tiny, which obviously are not always the easiest to lyse, where before and after we get the same amounts of protein. So if you can forward, Vic. If you can forward again. Again, just looking at our size and our ratios just for the cells that we can grow in the optimum growth from the 125 through the 7 liters. And as Tara did say, the low evap caps, we do have that for 125s. We are working on that for other sizes. And again, we are looking to optimize. This is an Inforce shaker where we optimize density, 18 1.6s, 12 2.8, and you do need a heavy load. So 35 liters, you do need the heavy drive for the Inforce or other shakers that you're using. If you can go to the next slide, please. So one of the things for larger cultures, if you are doing it, we do have the inversion and bidirectional transfer caps transferring to bags and to bioreactors. The other things people also use these for is feeds. So we see a lot of people using it for acid bases that they can use our inversion or bidirectional on their larger scale, 100 liter, 200 liter, up to 1,000 liter. And then for AAV, where you have a complex of DNA plus PEI, we see a lot of people use our inversion, and the inversion caps give about, you can take 3 to 5 liters and do it in less than 3 minutes. If you can scroll to the next. So again, we just showed here just a setup with a wave bag or with a 5 liter. So if you can scroll to the next. And then final is clarification and purification. If you can scroll forward, Vic. So we make a long drip. We have a 25 micron, and then we have a final filtration. So if you can scroll forward. So our rapid clear filter plates, we're going to go, we showed data before. If you can scroll forward. We have a 25 micron that you can add resins to. So if you want his tag resins, different resins, we can add that. And then Vic, if you can go forward. And then just a number of different plates. Please come look at what we have for SBS format. Can you scroll forward, Vic? And then please flask us anything. Thanks to Jeff Barnes and the marketing team. We're here for questions. We do have our FASs and our technical team. Alberto Estevez, who's on, Joseph O, who's back in the East Coast. And please reach out to our sales team, and we're happy to help with any of these processes. And again, this takes teamwork. It doesn't all happen in a vacuum. So thank you to the Thomson team, both the engineering and the overall team, and Tara. So please come visit us on our website. We're open to collaborate and discuss things that are needed. So Vic, would this be a good time for our question and
Q&A?
answer?
Yes, sir. So please drop your questions. I do see there's a lot of questions in the Q&A section. So if Sam, do you want to just
Sure,
pick
I'll
one and
take a
then
look.
yeah.
Let me just pick one for, sorry, I apologize, it's just loading. Here's one. Have you guys characterized oxygen transfer rate in your different flasks and plates, and how do they compare? So from Sam Siles. So what we've looked at, Sam, is we have looked at OTR, but we also look at geometry that's going to give the same density the same volume. And what we've done more than just an oxygen transfer rate is look at viability and VCD. Everyone is running different cells. So one of the issues that we have is a CHOK1 from, say, a Pfizer is not equivalent to a CHOK1 from Genentech. So what we do is we've looked over multiple beta testers to look at what speeds and times and volumes will work, and that's how we came up with it. Oxygen transfer rate is just one component. We look at components as OTR. We look at cell media and cells as the components that are needed, that make up what's needed. Alberto Estevez, let's see, can multiple protein complexes be expressed in the same system, Tara?
Thanks, Sam. So, yeah, that's something I didn't get to talk to in the constraints of the time. But that is a huge application space for the insect cell system because it is much more efficient to use baculoviruses to bring in multiple genes at once versus using multiple plasmids. It's much more efficient. And so using systems like multi-bac or BEVS or big bac can actually help you express. I think some of our customers are doing up to 15 different individual genes to create a large complex.
Okay. What system and expression systems is using for MOI determination?
So we use our baculovirus titering assay, which is a infectious units assay to determine our titer. And then we do the calculations for the multiplicity of infection based on that. We really prefer a low MOI strategy where using an MOI of 0.1 allows you to infect about 10 to 15% of your initial cell culture. What happens is overnight, the cells that aren't infected continue to grow, and the cells that are infected produce your next round of virus. And so you then use the power of the insect cells in the culture itself to spread, do a second round synchronous infection of the rest of your culture, which really drives up your infection densities without having to add large volumes of virus. If you want more information on that, I can explain or send you some papers on how we developed the titering assay and how that relates to determining your MOI.
Yep. And another question that we have is on this, can you make GMP grade AAV? I mean, I know the answer, but Tara, I'll let you answer that.
We can. And I did respond to you in the chat too. If you want to follow up at info@expressionsystems.com, I can give you more details on that. But the short answer is yes.
Yeah, and then another question for us is for what shakers can be used. You do need heavy drive. The only two that we see today that can take high volume, if you're doing 35 liters, I mean, Inforce has worked with us, Cooner has worked with us. Those are the only two shakers currently, if you're doing a high weight, that can do that. I mean, Tara, I would say in your lab, you agree, your Inforces work well, your Cooners work well for that.
Yeah, the biggest challenge we have with weight size is that nobody's comfortable doing 6 liters, even though they know that you can do 6 liters. They like 5 liters better. There's like this, I don't know, superstition around it. And so there's a couple more questions. Like one was, how would you recommend for determining virus titer in-house? So we sell our baculovirus titering assay. That's one way. You would need a flow cytometer, which can be limiting for some. We have a service that we can do it for you. And then there's other methods depending on, and this is why I like to talk directly to customers. If you have a cell counter that can look at diameter and swelling, we can help you calculate based off of that. There's a lot of different ways based on your available technology to help you determine a virus titer. I see the question also about what's the best solution to prevent contamination. Good aseptic technique. We really don't advise using antibiotics in your culture because it can mask low-level contaminations, it can actually negatively impact the mitochondria, and so you don't produce as much protein in the presence of antibiotics. It's a really, really good aseptic technique. Using the transfer caps that Sam was describing is a really good way of moving things from one area to another and minimizing operator handling. We really like those to prevent contamination when you're doing transfers.
So you've eliminated your bottles, Tara, and you're just using the transfer caps instead of a glass bottle with an assembly and all that. That's made it easier on you guys.
Well, you know, everybody's got their own techniques. We still have people sticking a transfer pipette into the bottle, but again, you know, a really practiced user has zero problem with that. A new trainee, a little bit more risky. So
We try to make it simple, Tara. And then also SF9 mammalian expression system, any on cell conditions. I think you can see Tara and I produced a paper last year on agitation, speed, temperature, and humidity. You can find that on our website, just on everything that you can do with, you know, right, Tara, that's still applicable. We're going about 135 RPM agitation on a 1-inch orbit, temperature. Do you have a
27.
temperature suggestion?
27. Don't let it
and
go over 28. It's better to go a little low than a little high. We also have, if you go to our website, if you click on the individual products, we have instructions for use. And so if you click on the media or you click on the cells, you'll get instructions that kind of spell that out also.
Okay. I think that's it on our side. This will be posted on Thomson's website. Again, if you have suggestions for things, let's see, there's another in the chat. You know, is it mentioned that some proteins express better in advanced CD? Is it just best to switch to expressing in advanced CD, Tara? Do you like advanced CD versus, or do you SF, or is it,
I
or is
like
it
the advanced CD. I
screen
think
as
it's a
possible?
great media formulation. It's really productive. It is more expensive though. So I know that some of our more cost sensitive customers, or those that are more about getting just some small amount of protein as quickly as possible versus really optimizing how much expression, it might be more cost effective for them to use the ESF 921.
Yeah, and Tara's welcome to connect with you on that. You know, look, just in essence of time, please contact us back, and we'd love to work further with you. Tara, thank you so much for the excellent presentation. Always fun working with you.
Thank you,
And
Sam. It's great.
please look, yeah, please come to us. We are looking to do some other webinars, and know that there's always new information, and appreciate everything you guys for you joining us today, and look forward to speaking with you and our team and Tara's team in the future.
Great. Thank you all so much.
Thank you all. Bye-bye.
Bye.
Bye-bye.